Let's remind ourselves of the central dogma of molecular biology. The central dogma of molecular biology states that DNA makes RNA, makes proteins. The process by which DNA is copied to RNA is called transcription, and that by which RNA is used to produce proteins is called translation. Today we are specifically dealing with translation. mRNA formed in transcription is transported out of the nucleus into the cytoplasm to the ribosome, which is the cell's protein synthesis factory. Here it redirects protein synthesis. Messenger RNA is not directly involved in protein synthesis. Transfer RNA, or tRNA, is required for this. The process by which mRNA directs protein synthesis with the assistance of tRNA is called translation. Another good point about translation is that prokaryotic translation can happen almost simultaneously alongside transcription. This is because there is no nuclear membrane within prokaryotes. Instead, the mRNA leaving the chromosomes can almost directly be in contact with the ribosome in the cytoplasm. This makes the process much more shorter and quicker. The process of translation requires many different types of molecules, including the ribosome, smaller and larger subunit, tRNA, the release factor, mRNA, and there are three stages of translation: initiation, elongation, and termination. The length of mRNA molecule varies. The average length for mammals, or eukaryotes in general, is approximately 2,200 nucleotides. This translates to approximately 730 amino acids in the average polypeptide. Only certain genes in a genome need to be expressed depending on either cell specialism or the environment. Therefore, not all genes are transcribed and translated. So, for example, if a cell needs to produce a lot of certain protein, for example, beta cells in the pancreas that specialize in secreting insulin to control blood sugar, then many copies of the mRNA are required. The genetic code is the set of rules by which information encoded in mRNA sequences is converted into proteins, which are amino acid sequences, by living cells. Codons are a triplet of bases which encodes a particular amino acid. As there are 4 bases, there are 64 different codon combinations. 4 times 4 times 4 equals 64. The process of translation can be summarized by the following steps. Initiation. Firstly, mRNA binds to a small subunit of the ribosome. The small subunit of the ribosome moves along the mRNA molecule in the 5' to 3' direction until it reaches a start codon. Start codons are usually, in most cases, AUG, U being uracil as it replaces thymine in mRNA, or RNA in general. A molecule of tRNA with its amino acid is complementary to the start codon, which is UAC, and it binds to the P site of the ribosome. The large subunit of the ribosome binds to the tRNA and the small subunit, and they move along the mRNA molecule. A second tRNA with amino acid attached, complementary to the second codon on the mRNA, then binds to the A site of the ribosome. The amino acid carried by the tRNA in the P site is transferred to the amino acid in the A site, as a consequence of the ribosome catalyzing a new peptide bond. This is known as a condensation reaction. The growing polypeptide increases in length. The ribosome moves one codon along the mRNA in a 5' to 3' direction. The tRNA in the P site is moved to the E site and then released, and then the tRNA in the A site is moved to the P site. So think of it kind of like a train. Another tRNA binds, complementary to the next codon on the mRNA, and it binds to the A site. And this keeps going on until a stop codon is reached. When a stop codon is reached, the translation is stopped. A release factor attaches onto the A site, and the polypeptide chain, which is now a protein, is released. The ribosome complex disassembles, ready for using or translating another mRNA molecule. There are two types of ribosomes found within a cell. Free ribosomes found in the cytoplasm synthesize proteins for the nucleus, mitochondria, chloroplast, and peroxisomes. Um, Free ribosomes are known to usually produce intracellular proteins. Membrane-bound ribosomes, however, are usually known to create proteins for the plasma membrane, secretory vesicles, endosomes, and lysosomes, mainly lysosomes. They are known for producing proteins that are usually secreted, such as insulin. There are four levels of protein structure. Whatever level a protein conforms to is determined by its amino acid sequence. So it's part of either primary structure, secondary structure, tertiary structure, or quaternary structure. For primary structure, the order or the sequence of the amino acids of which the protein is composed gives it the polypeptide primary structure, and it's formed by covalent peptide bonds between adjacent amino acids, so amino acids that are next to each other, and it controls all other subsequent levels of the structure. The secondary structure has the chains of amino acids fold on themselves, and they're held together by hydrogen bonds between non-adjacent, which means not next to each other, amine, which are nitrogen-hydrogen bonds, nitrogen hydrogen groups, and carboxylic carbon and oxygen groups. So the hydrogen bonds, they provide a level of structural stability, and we know this because hydrogen bonds also do the same thing in many other molecules, and these consist of fibrous proteins. Tertiary structure is when the polypeptide folds and coils to form a 3D shape, which is usually very complex, and it is caused by interactions between R groups, which are hydrogen balls, disulfide bridges, ionic bonds, and hydrophilic and hydrophobic interactions. Tertiary structure may be important for the function. So, if, for example, the specificity of active site and enzymes, and these are usually globular proteins. For quaternary structure, the interaction between multiple polypeptides or prosthetic groups causes this structure. A prosthetic group, in case you guys don't know, is an inorganic compound involved in a protein. So, for example, the heme group in hemoglobin, and these consist of both fibrous and globular proteins.